Hello children, the topic is molecular basis of inheritance. We are learning DNA. Stands for deoxyribonucleic acid. It is a complex organic polymer made up of nucleotides, 4 nucleotides linked by phosphodiester linkage to form a polynucleotide, 2 polynucleotides showing anti-parallel arrangement, linked tightly with each other by forming base pairs between purines and pyrimidines, and then they coil packed with histones in eukaryotic cells, giving a beaded string-like appearance. It is a genetic material already concluded by conjugation experiment, transforming principle, and transduction experiment confirms that DNA itself is a genetic material which can penetrate the living cell, incorporate and allow the production of replicas. Now what replica is? It is the ability to produce its own similar copy. To understand the autocatalytic property under the heading replication in DNA, we have to take help of experiment conducted by Meselson and Stahl. They proved, because there were hypotheses, a parental DNA molecule will remain as it is and it will produce another new copy. Means old DNA, parental DNA remains as it is and it is producing a daughter DNA which is having a new DNA molecule. Later, when Meselson and Stahl proved that parental DNA unwinds, each strand of parental DNA acts as a template to synthesize new strand. So, resulting new DNA molecule will be having one parental strand and one new strand, and both come together, form the base pairs and then coil. So in every DNA molecule, the parental DNA is preserved, which will help in synthesizing new DNA. Like this, it travels from generation to generation. That's why it is a base of inheritance. To prove semi-conservative, semi referring half old, half new. Conservative, the old nucleotides are conserved in the form of new. So parental DNA is helping as a template to arrange the new nucleotides which are complementary. So each newly produced strand is matching with one of the parental strand. This is how the replication is confirmed as semi-conservative bidirectional process. Let us study Meselson and Stahl's experiment. You have to listen very carefully now. They took E. coli, Escherichia coli, the most friendly bacteria which are harmless and symbiotic to human population. They cultured E. coli in a petri dish containing a medium of 15N. It is a heavier isotope of nitrogen. As you already studied in reproduction topic, bacteria can reproduce by binary fission. Means one bacterium divides to give 2, 2 divide to give 4. So when you are providing a nutrient medium, they undergo fission at the rapid rate, and during fission, whatever DNA they are having, they have to replicate it and they have to give equal quantity of DNA to the resulting daughter cells. So reproduction in bacteria is nothing but mitotic cell division where DNA undergo replication. Here in that nutrient source, the availability of nitrogen is in the form of 15N. It is a heavy, dense isotope. So in newly produced progeny of E. coli, that 15N incorporated in the nitrogenous bases like adenine, guanine, cytosine, thymine, having nitrogen with 15N. So from this first strain is generation, Most of the bacteria in which 15N incorporated in their DNA. So some of the bacteria from this first strain, they transferred to another petri dish where nutrient medium containing 14N. They allow bacteria to grow. So 14N is a precursor for synthesis of nitrogenous bases. So now they have two culture medium. One culture medium having 15N, another culture medium having 14N, and both the cultures, they allowed for centrifugation, keeping some bacteria aside. During centrifugation, they added a medium, cesium chloride, the more denser medium they have provided. When the centrifuged content is taken in a test tube, the first sample collection, they showed DNA pellets at the bottom because 15N is heavier. Second strain, they centrifuged in the same cesium chloride medium. There they saw DNA insoluble remained slightly upper in the test tube, not settled at the bottom. So this DNA is having one strand of 15N and another strand contain 14N. So from the second generation, the bacteria are kept aside. So these bacteria again, they grew in a petri dish containing same medium 14N. And third generation's bacteria, they subjected for centrifugation with the same medium adding cesium chloride. So when they collected the centrifuged content in the test tube, there some DNA are at the quite upper level, some DNA in the middle. So upper DNA they observed, they contain both strands with 14N, and in the middle whatever DNA was there, it is with 15N and 14N. So there are three samples. First sample, centrifuged content, DNA at the bottom with 15N. Second, when the bacteria transferred into the medium containing 14N, hybridization of DNA occurred, and they are slightly found in the middle of the test tube, lighter. One strand heavier with 15N and one strand with 14N. 14N is lighter isotope. So they are not at the bottom, they are in the middle. When the second generation bacteria continued to grow in same 14N, some DNA are quite at the upper side with 14N and some are in the middle. There is no DNA found at the bottom. So newly synthesized DNA incorporated with 14N, they become lighter, and those who remained with the parental DNA, they become slightly heavier, not completely heavier, because there is no source of 15N. By this experiment, Meselson and Stahl proved that replication in DNA is semi-conservative. So when this replication starts, last year you have studied cell cycle. It is a lifespan of the cell. Maximum time the cell will spend to prepare for the division. That preparation phase we call interphase. In interphase, the first phase, a newborn cell starts growing, it is G1 phase. So whatever DNA they received from the parental cell, this DNA is producing RNA, and RNA helping in synthesizing the proteins. After G1 phase, it is gap 1 phase. The longest phase is synthesis phase. So here actual DNA replication takes place. So synthesis stands for replication of DNA. And soon after replication, G2 phase begins, because newly formed DNA molecules will code for proteins which is necessary for cell division by producing RNA. So G1 and G2 is transcription, DNA producing RNA, followed by translation, RNA coding for protein to polymerize amino acids into polypeptide, and between these two transcription, the entire DNA undergo replication. DNA producing DNA is replication. So when DNA replication occurs in S phase of interphase of cell cycle. With this, I'll conclude this session. It is experimental proof for semi-conservative replication of DNA. It is optional question expected for 5 marks. Thank you very much.